dear students, good morning. Welcome back to the last session of the mitosis concept. So we had completed interphase and M phase, including the cytokinesis. Of course, karyokinesis followed by cytokinesis. Result, two diploid cells. This class is very special because we are discussing some uncovered points regarding entire the mitosis. Means the one who given the term mitosis, who studied mitosis, in plant cells which one is better, in animal cells which one is better to understand the mitosis in laboratory mode. Apart from this, we are going to learn some application part in the mitosis. Then why we have to wait? Let us see. Let us see the first one. This term mitosis is given by scientist Fleming. Clear? But where was the detailed study was first taken by Schneider. Schneider is the one who detailedly studied about mitosis first. If we see in the laboratory under the microscopic examination, from the root tips of the onion as well as the root tips of the broad bean are the best specimens for studying. So, in case of plants, onion scientific name is Allium cepa, as well as broad beans. In these two root tips, so if we cut the root tips section and stain with some color, and if we make some slides just like smears and observe under the microscope, we can see different stages. Sometimes some cells are in the rest phase, sometimes are in the function. If they are in the division, definitely in the root tip of the onion meristematic tissue is present. It is always divided. So that is the reason on root tips will be taken for the study of mitosis. Somewhere you can observe the prophase, somewhere you can observe the metaphase, somewhere you can observe the anaphase, somewhere you can observe the clear cytokinesis or karyokinesis. Now, in case of animals, first of all, best study in case of animal, then I can say red bone marrow as well as base of nail. Okay, in bone marrow different types of cells will be there. They can produce RBC one side, WBC one side, platelets one side. More than 200 different types of cells can be produced in the bone marrow. That is the reason bone marrow is also called stem cell. Clear? And why base of the nail? If you see the base of the nail is the growing part. Always it is in the division. That's why hair, where the hair tip as well as base of the nail, always they are in the division. And also cells of the skin cells. Every day when we wash our face, when we bath, when we rub, so many dead cells comes out. Means once the dead cells comes out, they will be replaced by the fresh cells, new cells, so that always they are in the division. That is the reason three regions we can easily identify to understand the mitosis. In case of animals, they are bone marrow, base of the nails and skin cells. So, okay, this is what a kind of information. Apart from this, what is so important in this session means calculation part. So, before going to learn about some analytics of the mitosis, one should understand that ploidity. What is haploidity and what is diploidity? Haploid, it is indicated by a single N, which means a cell which consists of a single set of chromosomes. Single set of chromosomes is called haploid condition. That haploid condition is indicated by single N. Whereas, what is diploid or diploidity? Diploidity means what? Double set of chromosomes. It is indicated by 2N. Generally, all cells except reproductive gametes in our body or any living organism body in case of a eukaryote. Okay, then all they are in the diploid state. So diploid state means what? Double set of chromosomes. So double set is indicated by 2n. Haploid is n, diploid means 2n. In case of human cell and human gamete. That gamete may be ovum or sperm. Understand? These are called reproductive cells. Clear? So the human cell is always in diploid. Human cell, example RBC, WBC, hair cells, neuron, muscle cells, nephron cells, everything, every kidney cells, whatever in our body, all they are in diploid. Even in the reproductive cells also diploid, but gametes, they are going to produce the gametes, they are haploid. They are having single set of chromosomes. So they are produced by meiosis. The first time you are listening in this session. Meiotic division is a reduction division which makes the number of chromosomes into half. So that diploid become haploid by meiosis. About meiosis in the next session we will discuss, guys. Entire this session belongs to only mitosis. So this is diploid. A diploid cells gives diploid, two diploid cells, right, by mitosis. All vegetative cells are divided through mitosis. Reproductive cells are divided through meiosis. But here haploid and diploid. Haploid is can be seen easily and very frequently in formation of gametes. Other than gametes, any cell in our body is absolutely diploid. In case of a human, diploid condition is 46 chromosomes. 46 chromosomes means 23 pairs. So that 2n is equal to 23 pairs or 46 chromosomes. In one cell of the human consists of 46 chromosomes. If it is in the interphase, entire this is in the chromatin. The condensation of the chromatin becomes chromosomes. How many chromosomes? 46 chromosomes or 23 pair. In human, when the cell comes into the prophase, all 46 chromosomes will be ready. After prophase, what will be continued? Metaphase. All these 46 chromosomes comes at the center by the help of 46 chromatids. Nothing but spindle fibers pair. Then one side 46, another side is 46, 23 pairs. The ratio of chromatids and chromosomes is 1:1. Now in the anaphase they are pulled and this side 23 pairs and that side 23 pairs comes. After karyokinesis and cytokinesis, one side 23 pairs along with the all cell organelles, other side is also same. Then two same type of cells are coming. That is the secret of mitosis. In mitosis, produced two daughter cells are as it is genetically similar, morphologically also similar. That's why they are called clones. When a cell provides two identical cells, they are called clones. So through mitosis, clones will come. The clones are also diploid. Here I would like to conclude with a point. In mitosis, produced clones or produced cells are genetically identical with the mother cell. They are always diploid. A diploid cell undergone the mitotic division, two diploid cells will come is the point you have to catch up. Next. This is a different case about the meiosis we will discuss later. This is a reduction division. Once a diploid cell, nothing but 46 chromosome having cell undergone, then 4 cells will come with the haploid. Understand? All these cells are having not 23 pairs, just 23. So number of chromosomes in either ovum or sperm is just 23, whereas other than this, any cell 23 pairs, nothing but 46. Clear? So upon this, some calculations are there. Let us see. So now I am representing one model of mitosis. You just follow. Catch the logic inside the problem. Don't think that the as it is problems may come. Clear? So understand the question that after 6 generations, of mitosis from a single cell. Half of the cells are found that are entering G0 phase. Here one cell is undergone 6 generations. One generation of a mitosis gives us 2. But here in the question mentioned 6 generations. So 6 generations means 2 power 6. That is 64. So 64 cells were formed after 6 generations. I repeat, one mitosis is equal to 2 cells. In the question they mentioned 6 generations. So 6 times mitosis happened. So 2 2s are 4, 4 2s are 8, 8 2s are 16, 16 2s are 64. So 2 2s are 4, 4 2s are 8, 8 2s are 16, 16 2s are 32, 32 is 64. So that's 6 times. So now half of the cells are found entering G0 phase. What is G0 phase? Here little concept is there. It is a cell cycle. This is G1, this is S, and this is G2. Later enter this is M phase. After M phase, 2 equal cells will come. Once a cell which is diploid enters the G1 for one G cycle, cell cycle, then this diploid go through the G1, S, G2 and M. 2 diploids will come. They are morphologically also same size. But if they want to divide again, they have to continue. So that 2 2s are 4 again. Again the cells again participate in the mitosis, 4 2s are 8 will come. But G0 phase is the phase through which cells comes out for function. Understand? They are not in the division. Once a cell comes through the G0, they never be divided. They meant for a particular function, that is a differentiation. What is that? When a cell comes through the G0, that phase is called differentiation phase. Now, this G0 phase is happened for half of the cells. Means 64 cells were formed in that by 2 is equal to 32 cells are in rest. Rest means out of division. They are not dividing. So out of 64, 32 cells are not dividing means rest of 32 cells remaining. Here you can see remaining half. Remaining half means what? This 32. In 64, 2 32s are there. One half, that is 32, is in rest. Another half, that 32 is undergone 3 more generations. So don't think that 64 cells are gone into the division. In the 64, half are in rest through the G0 phase called differentiation. That is called differentiation. Then rest of 32 cells are continued. How many times? 3 generations. So that 32 power 3. So 32 power 3 is equal to 256 cells. Now, so half of the cells undergone 3 more generations, then formed 256 cells. That is avoided division. The question is the total number of cells in that particular system. In that system, how many cells are there? Now 256 cells are there, but before the cells are not divided, they are 32. They are in the rest. We have to add them. So 256 plus 32 is equal to 288 cells are there in the system. The result of mitosis is 2. So that 6 times 2 power 6 is equal to 64. In that 50% are in rest, 50% are again divided. How many times? 3 times. So that 32 cube. That is 256. In the question they clearly asked how many cells are there in the system. Cells which are in the rest also present in that. Cells that are provided by the division is also in the system. So sum of the cells in the system including rest and division is 288 cells. Answer is 288 cells. Once a cell comes through the G0, that is called differentiation. Understand? Then if the cell, suppose 10 cells are there, some damage happened, some cells are missed, 10 become 5. Then how 5 become 10? Again they have to divide. For that again they are continued. From the function they are moving towards the division. then they call de-differentiation. Understand? Differentiation means a cell comes out of the cell cycle, then that is called differentiation. The same cell again from the work to division, then that is called de-differentiation. After that, again from the de-differentiation, it come back for the work second time, that is called re-differentiation. Here, three things are there: differentiation, de-differentiation, and re-differentiation. Understand? What is differentiation? A cell comes through the G naught from the cell cycle, away from the cell cycle, which is in function or rest. Whereas de-differentiation means what? It is from function to G1. What is it? Redifferentiation again from G0 to function. So this is our three different phenomena. Let us see another simple model. Second, another simple model. Let us see. 1024 cells found in a tissue. What would be the expected mitotic division number? So, if one diploid cell undergone mitotic, two diploid cells will come. No matter. Number of cells is 2. So here total number of cells are 1024. Then how many divisions occur? So 2 to the power of 10 is direct answer. Clear? So 2 to the power of 10 is 1024. 2 to the power of 2 is equal to 4. 2 to the power of 6 is equal to 64. 2 to the power of 10 is equal to 1024. So like that we have to answer. In the assignment you will see the different models. Only thing is you have to understand the data, what they have put in the question and what exactly they are asking. Okay guys, I hope you understand these logical thinking questions and all. In the assignment you will have these kind of questions by this session. But now I am going to visualize entire the mitotic division phase with a good animation. And later you have to practice this session and attend assignment. Thank you.
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads-on-a-string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as 2 sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach, by proteins in their centromeres called kinetochores, to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint, and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.